subaerial stem, long internode, runner. Prop roots, banyan tree. Free central placentation, primrose. Epigynous flower, guava. Ovary half inferior, plum. Unisexual flower, maize. Offsets. All bacteria having ribosomes and cell membrane, correct for bacteria. Mycoplasm lacks cell wall, true. Mycoplasm smallest cannot survive without oxygen, false. Membrane of protein classified as integral or peripheral, ease of extraction. RER contains on surface O2 surface of ribosomes. Origin of lysosomes, endoplasmic reticulum to Golgi body to lysosomes. True for ribosomes composed of ribonucleic acid and protein. Eubacteria resembles eukaryotic cell, plasma membrane. Outermost of plant cell envelope, cell wall. Allular stored protein. Alluroplast stored protein. Golgi complex function. Golgi complex function process and package protein and lipids. Endomembrane systems not correct. Plasma membrane is part of this system. Inclusion body store rule. Ribosomes location mitochondria and grana chloroplast. Incorrect for mitochondria. Enzyme of electron transport are embedded in outer membrane. 60s S and 40s 80s. Centrosome slightly away from middle of chromosome. Centrosomes होगा. Is submetacentric. Biomolecule. This reaction X and Y goes अलग जुड़ जाएंगे, साथ में हो जाएंगे, प्लस C डबल बॉन्ड आ जाएगा, लाइसेस. Exocrine A is pure of energy. BC in पता नहीं क्या लिखा है, एब्सेंस कुछ. Nitrogen base linked to the DHO of incorrect for enzyme formation of peptide proteins heteropolymer. Dietary protein source of non-essential amino acid, false. क्योंकि डाइटरी प्रोटीन्स आर द सोर्स ऑफ एसेंशियल अमीनो एसिड्स, क्योंकि बॉडी में नहीं बनते इसलिए बाहर से लेना एसेंशियल होता है, इसलिए एसेंशियल कहा जाता है. और नॉन एसेंशियल जो बॉडी में बनते हैं. इंट्रो डबल बॉन्ड एंड रिमूव H2O लाइएस. एनिमल वुड abundant protein collagen. X is न्यूक्लियो क्या कैटेगरी है, न्यूक्लियोसाइड कंपोजिशन यूरेसिल. Higher in human body, the earth crust, carbon. Primary protein structure, positional into of amino acids. Most abundant protein in whole biosphere, Rubisco. Introduction of double bond and removal of water, lyase. C double bond O, single bond and H is not a amino acid. Nucleotide of phosphate group, phosphoester bond. Phase. Resting phase is predominantly phase for division. Planetary resistance, xenocytic type and emission environment interaction. Phenotype resist, genotype and environment interaction. F1 selfing 1:2:1, tall homozygous, tall heterozygous dwarf. Pink and white variabilis, genotype phenotype 1:2:1. क्या लिखा है यार ये, and 1:1, 1:1 and 1:1 ही लिखा है. 90 dihybrid, 90% parental combination and 10% recombinant. Incomplete linkage. Autosomal recessive. Not example of incomplete dominance, Pisum sativum. Not autosomal linked disease, hemophilia. ABO blood grouping codominance. ABO genotype 6, phenotype 4. Function of biofertilizer glomus. Streptokinase myocardial infarction. Streptococci. GERD of illuminant and marshy area methanogens. Fix atmospheric nitrogen while free living soils. Azoospermum azotobacter. Biotechnology. Tetracycline resistance PBR322 cutting by BAM HII. DNA protein and RNA molecule isolation, proteinase and RNase mismatch. Plant cellless. Top polymer in PCR. Extinction of primer and on the template DNA. Heterologous host ROP. Production protein for reproduction of plasmid. ROP. Check progression of restriction enzyme digestion. Glycerate of growth. Reaction endonuclease sugar phosphate. Salt concentration land H2O less than 5%. Sea anemone and hermit crab commensalism. Topographic factors slope of earth's surface. Asymptote K equals to N. Species living in well defined geographical area is population. Amount inorganic substance present ecosystem preventing area at deviated period standing state. Amount inorganic substance present ecosystems per unit area at a given time period standing state. Successes in aquatic habitat forest to mosaic no number of bivalent in pancreatine stage in parent cell. Correct 24 chromosome in G2. The end of first meiotic division and chromosomes of chromatids. To the metaphase mitosis ER and the nucleus begins to disappear. Late prophase. Separation of paired homologous chromosomes begins. Diplotin. Meiosis 1 synapsis takes place. Zygotin. Bivalent of meiosis 1 4 chromatids and 2 centromeres. Synthesis of tubulin G2. Centrioles starts to move opposite poles. Prophase. Number any enzyme synthesis at M-phase. Segregation of homologous chromosome pairs occurs in segregation of homologous chromosome pair occurs in anaphase 1. Enter-kinesis stage between meiosis 1 and meiosis 2. Enzyme in crossing over recombinase. Protein synthesis G2 phase. Diplotin of prophase 1. Dissolution synaptomer complex. G2 में no duplication of centrioles. Bivalent vesicles tetrads pancreatine. Intervals between the mitotic phase and DNA replication G1. Centriole duplicate S phase. Move opposite poles. Chromosome moves opposite pole. Prophase of mitosis. Complex disintegrate of nuclear envelope marks start of mitosis metaphase. Crossing over between with chromatid and with stage of cell cycle. Non-sister chromatids of homologous chromosomes at pancreatine stage of prophase 1. Meiotic cell followed by cytokinesis leading to the formation of diet cell telophase 1.